Hi, I'm Joe Aslan from Oregon Health and Science University. Today I'm here with Kanika Jain, research faculty at Yale University, who's a finalist for the 2025 Brinkhous Early Career Award in Thrombosis. And I'm excited to talk to her about her research and what she's been up to. So Kanika, how did you get into cardiovascular vascular science before your recent role, and what kind of brought you to your project that you'll be talking about today?
Yeah, hi, thank you for the introduction. So, I've always been interested in proteostasis and understanding how proteins misfold and what can be the consequences of this misfolding. So, during my PhD, I worked on this in context of cardiovascular biology and understanding how the misfolding of proteins can regulate both adaptive and maladaptive responses. But when I moved to Yale, I was always curious to know whether this can happen without a nucleus in a cell like platelets. And through the years, we have deciphered that this response, which is known as the unfolded protein response, that gets triggered by the accumulation of misfolded proteins, gets activated in platelets and can play a major role in defining how platelets are activated, what are the functional responses, especially in the course of hemostasis and thrombosis. After that, I think we were wondering where next to take this project. And the one area of research that we then got excited about was neurovascular diseases with a focus on dementia and Alzheimer's disease. And we found that platelets have been usually the ignored cell types in Alzheimer's disease. However, platelets actually possess the biggest repository of amyloid in the blood. So, which got us excited, and then we started figuring out more about what happens to the platelets in Alzheimer's disease, which forms the focus of the current work.
Did you start looking at human samples or mouse models of Alzheimer's disease? Did you look at platelets specifically, or physiological slices from different models? Like where did you first start looking for your phenotype?
So Yale has a very wonderful Alzheimer's disease research unit. So we started collaborating with some of the clinicians there, and we talked to them about this project first, and they mentioned that it was a very cool idea, but they didn't know more about it to kind of guide us. And then we started looking at patient samples. So we got patient samples who were screened at the Alzheimer's clinic, and we looked at their platelets, which then led us to consider whether what our findings we could recapitulate it in the mouse models. But that's where we are working at right now.
That's really exciting. Do you think that your research will lead to a way to better detect or screen for Alzheimer's disease using platelets? Do you think this phenotype will come up as a precursor, or is it something pretty late into the disease?
That is an excellent question. I think one thing we can definitely say that a lot of changes that we are seeing in the plasma, and there has been a recent emergence of interest in plasma biomarkers for Alzheimer's disease, especially with all the new FDA-approved drugs which have been on the market in the last year or so. So we have been seeing that a lot of those findings are actually derived from platelets, and in fact the severity of disease itself we see is being correlated to what we see in the platelets, which opens up quite a few new areas of study, and it is also non-invasive. So we feel that that is a great way to take this project forward, where we can work both on the mechanistic side of things but also on the biomarker side of things with this project.
Oh, that's great. Do you see any similarities in other protein folding diseases in neurology, like Huntington's or Parkinson's? Do you think there's going to be any similarities there, or is this an amyloid-specific effect?
Okay, I am quite excited to answer that question because I am in parallel working on a Parkinson's disease project as well. And so, yes, I can say confidently that we are seeing a very distinct phenotype which makes both Parkinson's and Alzheimer's disease different from the perspective of platelets, but also there are overlapping features there. So it is a very exciting area of research for us.
What are you excited about for the next steps of the project after this meeting? Where are you taking your work next?
I think there are two major areas. Like I mentioned, one is the mechanistic side of things. We are now focusing on how the UPR, which is the unfolded protein response, how that can regulate the platelet changes that we see in Alzheimer's disease. So that's one active area of research. Plus, there are a lot of drugs that have been considered which can target the UPR. And we feel that that could be a very new direction of research to take, where we can target the misfolded proteins both in the brain and in the platelets by drugs which are approved for the inhibition or activation of the UPR response. The other side is obviously the biomarker side, where we hope to define more significant directions and see if platelets can become the more robust biomarker for Alzheimer's disease.
Do you see anything different about the activation state of platelets in Alzheimer's disease?
Yes, so we have been seeing that platelets are hyperactivated in the disease, and this is in line with what happens when there is a misfolded protein accumulation. It's kind of following up with our previous work, where we see that platelet hyperactivation occurs when there is a lot of protein aggregate that starts to gather in the platelets. It's also interesting because aging is a major risk factor for Alzheimer's disease, and aging is also known to cause platelet hyperreactivity. Although we have shown that it's non-linear, it may decrease with advanced age since there is an adaptive response that stems in. But with Alzheimer's disease patients, we are seeing a very interesting pattern.
Yeah, that's great. I'm really excited to see your presentation today, and I look forward to learning more about your work and just seeing where it goes.
Thank you so much. Thank you for talking to me.
Yep, thank you.